talking about what happens when we get sick. Now, I'm not talking about just a little sniffle, I'm talking about full-on cold or flu or even, God forbid, HIV. So, does anybody know the difference between getting the flu than getting strep throat? What's the difference? Michael?
The flu is a virus and strep throat is bacteria.
That's correct. That's what our whole premise today is going to be about. So you'll notice people in the movies, when they get sick, they don't look like they're really sick. They just look like they're all at peace with an ice pack on their head. This is me when I get sick, and I'm pretty sure everybody else is too. But that's definitely me. So right off the bat, you can tell a huge difference between a virus and a regular cell structure. What is one thing that you can point out to me that is a lot different than a regular cell about the virus? Randy? The organization and shape, that's correct, yeah. You notice these little sticky things outside here? They're called glycoproteins. You'll notice that the cell does not have those. Can anybody tell me why? So the glycoproteins are to attach itself to a regular cell such as this and go inside of it and infect it. And as you can tell, they're probably really good at it, so because they have so many. You notice another thing about this virus structure is that it has RNA. What do we have in our bodies? DNA, that's correct. So we'll look at the nucleus here. Do you see a nucleus over there? No? Okay. Viruses are not nucleated. They don't have a nucleus. And they have RNA and they replicate with RNA, not DNA, like regular cells like in our body. So this makes for a really difficult way to take care of it. It's very difficult to cure a virus, which is why we see these epidemics such as HIV, which is really life-threatening virus to have. And then there's common viruses such as the cold. Now, let's move on. So tell me a few common viruses. I've already mentioned a few, which is the HIV, common cold, and here's a molecular level of the HIV. I know it looks kind of pretty, but it is not. And then on this graph here, you'll see that there are several strains of viruses, of the HIV virus in particular, in all different parts of the world, which makes it difficult to find a cure for it because all of the vaccines, there are so many different strains of it that the vaccines cannot keep up. And we'll talk about why with this video here. This not possible. Okay. First video is how the HIV cycle works.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell, where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
So what can you guys tell me about that video? You guys know a little bit more about how HIV enters the cell and how it damages the cell after it replicates? Okay, will you guys be able to tell me this on the test? Okay, great. So, let me go on here. We're going to look at the entry of virus into a host cell quickly, and then
There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface, called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
Okay guys, so y'all see that it is a lot similar. This particular virus is not named, but it's a naked virus, of course, but you see how similar it is to the HIV virus, how that enters the cell. They both enter by attaching themselves with their glycoproteins onto the receptors of the regular healthy cell, and then they go in, they do DNA transcription through RNA, messenger RNA, and then they move out of the cell, and eventually HIV kills the cell, and then it just moves out and continues to replicate. Now, we're going to talk a little bit about mutations later on today, but that's the basic way of how viruses replicate within your body. So antibiotics do not cure viruses. Can you tell me why? I know it says it up there, but did anybody know already?
Viruses aren't bacteria.
Viruses are not bacteria. Antibiotics means cure for bacteria, and it's a viral infection such as the cold or the influenza virus is not bacteria, so it's not going to help you at all. So if a physician tries to prescribe you antibiotics for the common cold, what do you say? No, that's right. Why? Because it's not a bacterial infection. may even cause you more harm because it's going to weaken your immune system there, and you can get even more sick from it. So now we're going to break into groups and look at different types of viruses and draw out the virus cycle. Let's move on.